Hello, Database. Thank you for joining us. Great.
We have the lovely Francine Lejeune joining us.
Hey,
That's my
Francine.
wife.
Awesome, great. And the room is still filling up. Hello, Database. Thanks for joining us today. Would love to know where you're joining us from. Go ahead and drop your location in the chat. Always love learning where everyone's coming from today. And we're just going to give it about another minute or so. The room is filling up with our attendees, and then we're going to get started. I'll go over the ground rules for today's webinar, and then just briefly talk about the Hive Think Tank before passing it over to TM Ravi, the Hive's founder and managing director. And then we're going to get rolling with our incredible panel. This is going to be an amazing conversation today, so I'm going to try and keep it very brief. Awesome. Thanks, everyone. Los Altos, hello. Jan or Jan? I'm gonna get Jan. Jan. All right.
Good friend of ours.
Awesome. Hey, Kaiting from Sunnyvale. Hey guys. Thanks for joining. Where else is everyone joining from? Do we have any Australians in the house? Okay, give it one.
I can count myself as one.
Great. Okay, so room is definitely filling up here. Oh, Livermore, Santa Clara. Awesome. Great. Thanks, guys, for letting us know where you're from. Portola Valley. So we've got a lot of Bay Area folks. Okay, so I'm going to get started here. Ravi, do you mind pulling up the PowerPoint?
Yeah.
Thank you so much. Hi, everyone. Thank you for joining us, and welcome to the Hive Think Tank's webinar series. My name is Maddie Watt, for those of you who don't know me, and I'm the Senior Manager of People and Programs at the Hive. We have an incredible panel lined up for you guys today. We're going to be talking about personalization and healthcare with data and AI. This is being co-hosted with Darling Ventures, and it's in conversation with the University of Texas's MD Anderson Cancer Center. They are the largest cancer center in the United States, and that will be joined by Dr. Andrew Futriel and Dr. David Jaffrey. And these guys are the top of the top in terms of cancer research and technology and genomics and what is leading in that industry. So you guys are in for a really good treat today. And I want to do a special thank you to Darling Ventures, who has collaborated with us on a 3-part series in healthcare and AI. Today's webinar is part 2 of that series. So if you want to join us for part 3, you need to make sure to join our meetup group so you stay in the know, and I will drop that link in a little bit. So real quick, ground rules for today. Please be respectful in the chat. Please refrain from product plugs, and please don't recruit for your startup. Thanks so much. We love questions. To ask a question, please use the Q&A button, and that's going to be at the bottom of your screen. You guys will be able to see it right underneath the video area, and we appreciate you asking questions through that instead of the chat, because that way it's in one nice, neat place for our panelists, and it makes it a lot easier. So if I see you asking in the chat, I'm going to have to yell at you and make you use the Q&A button, okay? Thanks, guys. And then this session is being recorded. It will be available on our YouTube, and I'll be sending that link out later, and I'll also drop the link in the chat. And to follow along or contribute on social media, which we love, please use the hashtag HiveData and tag us at HiveData. And at the very end of today, I'm going to send out a brief event feedback survey. It takes 1 minute. If you have the time, it helps us customize our events catered to you and your needs, and we really appreciate it. Thanks so much. And then next slide, Ravi. Just want to say a special thanks to our sponsors here in a minute. So the Hive Think Tank is an ecosystem of thought leaders, entrepreneurs, and corporations. We are an event and content platform. We are bringing you events nearly every week, and you can find out about all of our events on Meetup. And we also publish thought pieces on Medium, and I will drop those links in the chat. Next slide, please. Again, thanks to our sponsors, and special thank you to Daniel Darling of Darling Ventures for collaborating with us. If you want to join the Hive Think Tank in terms of sponsorship, or if you want to collaborate for an event, please email me. I'll drop my email in the chat. And for next week, we would love if you joined us at our event. It's okay, it's probably at the end. We have an event next week. And Sorry, guys. That was my bad. I must have put the, there we go. I see it. Perfect. Sustainability action in our new world. So this is going to be an awesome event on sustainability and climate change. And we're going to be joined by a bunch of different thought leaders in this space, but particularly Rob Swan is the first explorer to ever walk the North and South Poles. He's also been honored by Queen Elizabeth II herself. So it's a really big deal if you are interested in sustainability, particularly if you are getting involved in climate change after all these wildfires on the West Coast. This is an event you definitely want to join. So I'll drop the link for that in the chat. And without further ado, here is T.M. Ravi, the Hive's Managing Director and Founder.
Thank you, Maddie. And in particular on this event around sustainability, in addition to the amazing Rob Swan, we also have Roberta Bauman. And Roberta is now at the ladies PGA, but prior to that, she was at Duke Energy. And think about sort of being the Chief Sustainability Officer of Duke Energy. So her insights here about what does it mean for corporations to have sustainability goals. And the notion of sustainability that we'll talk about in this particular event goes beyond environment and climate change, also social impact. So think about social justice, diversity, and inclusion. So it's going to be an amazing event. I hope you can join us. It's next Thursday, if I'm not mistaken. So welcome to all of you, data beasts, to the Hive think tank. A brief introduction. The Hive is a venture studio. We focus on data and AI-driven transformation of the enterprise and a number of different industries. In particular, today we want to talk about the last area, which is on life sciences and healthcare. We have collaborated actually with our moderator here, Daniel Darling of Darling Ventures, to do a company called Glide Health in this space. We're very interested in the overall areas of personalization of medicine, of digital health, of using sensors in this area, genomics, computational genomics. So if you're an entrepreneur, want to start a company, reach out to us. We'd love to engage with you. And so with that, I'd like to introduce my friend, our colleague, our collaborator, Daniel Darling. Daniel is the managing director of Darling Ventures. It's a San Francisco-based venture firm, very similar kind of philosophy to the Hive, kind of focusing on fundamental technology. They're also early stage investors. We have collaborated on multiple companies in addition to the one I mentioned, Glide Health. And the special connection that Daniel has to today's event is that Daniel is a member of the MD Anderson Advanced Team Board that connects MD Anderson to Silicon Valley. And so today's event is part of is one of a three-part series. We did one a month or so back around affordability in healthcare and medicine. And today we'll be talking about personalization of healthcare and medicine with data and AI, and expect to hear and see a notice about an event around preventative healthcare sometime in the next few months. So with that, I'd like to hand it over to Daniel.
Thanks, Ravi. Welcome, everyone. Thanks for joining us today. Very excited to be approaching the topic of personalization of healthcare and medicine with data and AI through the lens of MD Anderson Cancer Center. Cancer is arguably the most complex and widespread disease that we face. Each case is totally unique and in desperate need of personalization. So if we can solve for personalization in this complex environment of cancer care, we can definitely solve it in the doctor's office. So really fascinating lens to be approaching the topic through the lens of MD Anderson and cancer care. Now, MD Anderson, for those who don't know, is a real global leader in the cutting edge research and advanced care in our fight against cancer. Some quick stats. So there's 22,000 employees who focus purely on cancer over at MD Anderson. They touch 1.5 million patients yearly at the institution. They do over 1,400 clinical trials every year in their research. That includes also doing 13 million lab tests annually. And in terms of how much effort and capital they put behind this and advancing the field, they invest $1 billion a year in research. So we're talking an institution with major global impact and major reach. What's great is that today we have the pleasure of looking at the personalization of healthcare medicine through the eyes of not only MD Anderson, but its boldest innovators and leaders, Dr. Andy Futrell and Dr. David Jaffrey. Dr. Andy Futrell is the chair of genomic medicine at MD Anderson. He's published over 230 papers at the institution, and he's co-leader of multiple frontier initiatives at MD Anderson, pushing the envelope for personalized healthcare and personalized medicine. This includes Translations Research Accelerator, the Genomics Laboratory, the Apollo program, which stands for Adaptive Patient-Oriented Longitudinal Learning and Optimization, a mouthful, but we'll get into it later into this session. And he's led multiple breakthroughs in genomics at the institution, and very, very passionate about the intersection of healthcare and data and how he's leading multiple initiatives there to drive that whole collaboration forward. His sparring partner and colleague is Dr. David Jaffrey. He's the Senior Vice President and Chief Technology and Digital Officer at MD Anderson, and he's the first person to hold that position at the institution. He looks after the design and development and deployment and management of all the enterprise-wide tech and infrastructure that goes into the institution. So really fantastic perspective to have him on the panel, and also looking at not just internal initiatives, but also those that deal with external partners and strategic partners to help them go forward. He's been the President of Technology and Innovation at the University Health Network and Princess Margaret Cancer Centre in Canada, and he led their digital transformation there. So long history of experience in this space. On top of that, he's also a professor at the radiation physics and has authored more than 275 peer-reviewed papers in that space. So needless to say, we're in fantastic company here for this discussion, incredibly qualified and experienced individuals to start to look at this intersection of data and AI and the healthcare industry, and in particular around personalization of it. So Andy, David, very warm welcome to you both.
Thanks for having us.
Yeah, it's great to be here.
Fantastic. So why don't we start off? So why is personalization so key to winning MD Anderson's fight against cancer?
Well, I can try to tackle that one to start with. I mean, I think you hit on it in the beginning, Daniel. It's what you're dealing with is an incredibly heterogeneous disease. And even if you think about it in what is now probably the most simplest context, which is just think about the DNA, the genome, that we've been studying for the greater part of 20, 25 years with some degree of detail. Even in an individual patient, you have multiple components in play. You've got the germline, that is the sort of the deck you were dealt from mom and dad, which makes us all slightly different from each other, and all that imparts in terms of the genotype, phenotype sort of correlation. Then you have the tumor itself. And what we've learned over the last, again, 10 years or more is that instead of a monolith, a cancer in a patient is a collection of species, tribes, with different levels of activity, different behaviors. And when you put pressure on that system, what you get is the common phenotype of patients developing resistance to therapy. This is a manifestation of the heterogeneity in the underlying system. So even if you stop there, that's a very complex problem to solve, because each tumor at the base per base level is completely different from every other cancer. And within that mass, there's multiple species. The third leg of that heterogeneity, just based on purely DNA in the first instance, is now we've learned that not only is it the genome that you were born with, the genome acquired by the tumor cells, but also the genomes of all those organisms living on us and in us in the context of the microbiome that are having, ever more remarkable sort of influence on response to therapy, to risk, to outcome that I think we have to take into account. So you've got this complex interaction of genotype, phenotype, even within a single patient. And that's not even thinking about exposure, lifestyle, diet, all the other things, which we know from just purely large amounts of both trial and empiric evidence have huge effects on outcome. And so I think that begins to paint a picture of, even with the best, smartest people in the world, you run out of runway in the brain space to begin to think about the numbers of correlations and the impact of the interactions, not only in a point in time, but also over time. Because that is the other key factor we largely ignored because we wanted to understand at least snapshots of the system. Now what happens when you put that train in motion? Over time, under therapy, watch things develop, evolve. And so you've got dimensionalities there in terms of complexity of heterogeneity and interaction that are really difficult to come to grips with. And it's one of these things that drives the entire system and drives resistance, drives refractory disease, drives risk, and we just have to get to grips with understanding it. The technological barriers are getting lower, but we still need to think about the organization of all of that data. And so I think that's sort of where we start from thinking about, the capability to personalize or precision medicine in the context of cancer. It's a knowable space, even if you just stick to the genomes.
I think one of the things that makes it really clear and brings it really clear to us is that at a cancer center, we treat the one person the same as the next person to the best of our ability, yet we get a different result. Mm-hmm. And to me, that just sort of says to us that there's got to be some clue. There's got to be some nuanced difference that we can somehow detect. And what we're learning is it could be several clues that have to be brought together to figure out why that patient responded differently than the previous patient. But every time a patient comes in the door, we get reinforced that it's a variability, and we're trying to understand what's the cause of that variability, because if we could figure that out, we can make a better choice for that patient. And that's that personalization that we're so hungry to get to.
And where are we in that journey? How personalized is medicine and care today?
Well, I think we've made huge strides, and it's taken some interesting, at least to me anyway, twists and turns over the last let's call it decade. Certainly, we've gotten to grips with the individual character of a tumor at the level of what genes are mutated by defining the list of genes that seem to be important in cancer, the quote unquote cancer genes, and developing drugs for very specific recurrent stereotypic mutations in those genes. So we do a pretty good job now of being able to essentially genotype, if you will, cancers and picking therapies off a shelf which have been sort of designed to act upon that particular genotype. That's sort of overselling it probably just a little bit, because there's complexity in, it's not just one gene that's mutated, it's usually multiple. So context matters. We're beginning to sort of understand that context space and think about combination therapies or hitting, multiple legs of the stool at the same time. The other piece that sort of happened in the last decade in terms of progress in personalization, seen through a different sort of lens, is this idea that you already have a system in your body that's geared to sort of deal with foreign invaders, for lack of a better way of putting it, and that's your immune system. And it's incredible strides in understanding the interactions between the immune system, the host, if you will, and the cancer as invader, and being able to tweak that system. And that is the ultimate sort of in the body personalization system to go after a particular cancer in that individual patient with memory down the road about how to treat it if it comes back. So those two prongs, I think, have really led us to think about in a very sort of tumor-centric way. How do you begin to think about personalizing medicine? I think there's a long way to go in the context of thinking about moving from tumor-centric, I would call, to more of a patient-centric view. And again, taking into account all these other variables, the comorbidities, the lifestyle exposures, et cetera, that I think can influence that particular bit we already know about. So I think we're moving definitely in the right direction, some huge advances in being able to leverage targeted therapies, immune therapies in patients. But I think we've a long way to go in terms of making those really active in the majority of patients.
So really the immune system and immunotherapy is that holy grail for personalization in your eyes?
Well, I mean, I think it is a huge leg of that particular triumvirate of sort of, surgery, targeted therapy, immunotherapy, radiotherapy would be the other sort of the fourth pillar, if you will. And we're to the point now of actually thinking about how do you leverage all of those things in combination to let the immune system do its best job. What is that right combination or even the order with which you would use those particular therapies in the context of getting the system, that is the patient's response system, to actually target and eliminate the majority of the cancer in the majority of patients. And that's a large amount of ongoing work to understand, how do you treat the system, what order, what dose, what schedule you need to use for these particular sets of agents in a particular genetic context. So it's very difficult to escape the genetic context because that drives the immune system as well. So it's the interplay, which is kind of why we're here to talk about how you get to grips with that complex interplay.
Mm.
And the
Sounds
key
like
is there's just so many aspects to this, right? Andy pointed out, I mean, when you bring a patient through for care, and we think about it from a multidisciplinary perspective, bringing all these aspects together just sort of highlights some of the complexities that are present. So what's the right sequence? What's the right state that the patient's in? Is their immune system going to be responsive to the intervention to go against the cancer? I mean, is each cancer going to respond to that immune response? And so the dimensionality is really remarkable. And trying to figure out how to optimize that is interesting from a measurement point of view, but from a computation point of view as well.
Indeed. And a key first step in this personalization seems to come to the sequencing of the genome and the sequencing of the DNA and all the rich data that provides. Are we moving towards a future of more continuous sequencing or maybe even sequencing at birth?
I think my personal view is that's inevitable bit of technology development and lowering of costs that will eventually get us, this is just routine. I mean, it's a blood draw, essentially. I think the other dimension, which I think you hinted at there as well, is what we've also learned, and we've actually begun to build systems just to direct and go after, is it's not enough to do it once. Particularly in the context of the tumor, but think about the microbiome as well. You need to really think about multiple looks into that system over time and into therapy. And the analogy that I always use is you don't do a single CT up front and then try to understand the complex biology by never imaging again and just relying on that one. So, you make the argument that molecular information and molecular circuitry in a cancer is important, or the molecular circuitry and the interaction space between microbiome and cancer is important. Why wouldn't you want to know what's happening once you start to poke the system with a stick? And I think that's what we've purpose-built platforms that are there to do nothing more than think about identifying patients, thinking about their schedule of procedures. How do we get there to get the sample? How do we go back and get the sample at progression? How do we go back and get the sample at second progression? And do the deep molecular measurement space over time, as well as just the snapshot view. So I think we have to sort of develop the dimensionality, think about this as an ongoing inquisition into the system itself, I think.
I think this is one of the most interesting things. One of the reasons I joined MD Anderson when I did a year ago is that, I mean, this is not the standard of care paradigm. This is about learning as much as we can in caring for a patient and to try to understand the trajectory, where they are in that trajectory, and then how do we optimize in the intervention. And the scale of that problem is really substantial from a computing perspective, from a data collection perspective, and then even from an optimization perspective. And to me, this is a really key element. If you think about the time course of treatment and the time course of disease, we have sort of in a classical medicine way, we take a snapshot, we prognosticate that there's an intervention, what's the likelihood of the outcome. But these are dynamic systems, complex dynamic systems. And how do we wrap our minds around that? How do we get the right measurement to give that the appropriate, most effective treatment for that patient? And it's a fascinating problem, I think.
Indeed. And David, you've recently joined MD Anderson, taken the helm in charge of this with the CTO position. So give us an idea of what are your plans for the institution, how to unlock some of this value in this data. The scale of data that you guys handle is really mind-blowing. So give us an idea of what that looks like and how you're approaching it.
Yeah, I mean, it comes from our leadership. Our CEO is extremely enthusiastic about understanding how we use data to advance the mission, which is to end cancer. And it was a couple of years ago that I first met Andy, and you've heard him talk about already about his enthusiasm of understanding the disease, but the patient and also the intervention in detail. And so we really struck it off and aligned in our thinking. And so we took that thinking and we said, okay, if I'm in charge of IT for the organization, how do we build an organization that's built to really leverage the observations that we make in the clinical context, in the research context, in the laboratory context, and thread them together so that we can really get critical insights on a discovery basis, but also on a care basis. And so we've started down a line of work now, which is to really change the way we think about the flow of data in the organization. It's been very exciting over the past 6 months or so. We've been making a lot of progress and getting the organization excited about how we manage data, how we think about the flow of that data, how do we know where it came from, the quality of that data. Because we're convinced that if we fumble the ball in handling that data, the nuances that we will lose will undermine our ability to predict the personalization, to understand the heterogeneity. So it's kind of like, to personalize, we have to industrialize the way we think about handling information in the organization. And because it's little differences that are critical. And that's what we've learned. We've sort of simplified the description of cancer in the past. And we learned that solving a simple problem isn't going to actually solve the problem we need to solve. And so escalating the complexity, capacity of the organization is key. And as Andy's described so nicely, there are so many dimensions to this problem.
I mean, I think it's sort of, get back into this sort of this tension between knowing that numbers matter. I mean, if you start talking about even things as simple as, you know, variability in genotype in the population, you know, it's a bell curve at the end of the day. So the majority of us have most of our variation in the middle of that curve. To really understand what tiny role any one piece of that variation may play in a clinical phenotype response to therapy. You have to have large numbers to get the signal to noise ratio to tell you that that's real signal. You can nibble at the edges. On the other hand, to get really deep on every individual patient, and one of the things that David first said when I met him was he likes measurements and more measurements. Well, how do you get into more measurements and more measurements? And that does require this nexus of knowing that you have to learn from every patient, but also industrialize the capability to do that, and also be able to handle that data on the back end so you actually maximize the value of every data point from every patient. And that's the challenge, I think, at the end of the day.
I mean, one of the things that's really interesting too is if we have a different outcome, we want to make sure that we treated each patient the same, at least. We want to really understand the consistency. So remarkably, treating patients with a very high quality and consistency is a good thing for the patient, but it's also the way we will better understand why one patient responded differently than the other. Because if we treated them really consistently, but we still see a difference, maybe the difference is in that subtle genome measurement that Andy just made. Maybe it's lying there, but we can now see it because we treated the patient in a very high quality consistent. So the alignment between the quality of care and the consistency and our ability to discover, if we bring it all together, I think it's really a kind of virtuous cycle.
And when you talk about the industrialization of that process, it's about achieving that consistency, right? From your perspective. And seems one of the amazing initiatives you guys are doing is the Apollo program. You're both leading that initiative. Maybe you can help us understand what that looks like as an implementation of what you're talking about.
Yeah, so the Apollo program was born out of a really sort of simple need, which I think most good ideas are born out of really simple questions sometimes. So again, it gets back to this idea that the most common observation in oncology is you treat a group of patients who ostensibly are the same and you get different outcomes. And how do you get to grips with that? Well, we've talked about heterogeneity. We talked about that, you know, there's inherent dynamics in the system once you start to put pressure on it. That's a problem. So how do you deal with it? The way you deal with it is to purpose build systems which allow you to measure over time. With certain degrees of accuracy. And so we took upon, you know, several years ago in the context of the moonshot effort I lead to sort of Okay, we need to build the plumbing. So how do you do that? And it's, it's not particularly glamorous stuff. It's really sort of, patient scheduling, consents, being in the OR or the IR suite or whatever to collect the tissue, and then putting in quality controls before you ever generate data. And so one of the things that's plagued research is, and I come from this world. If I wanted to study breast cancer in the past, maybe I'd ring up, somebody I knew at a medical facility or a hospital or whatever, and I'd say, do you have any samples? And they say, yes. And they hand me a rack of tubes labeled 1 through 10. And I do stuff to them, I generate some data, and then maybe I worry about at the end about, well, what was the actual histopathology of the sample that went into tubes 1 through 10? And you get what you build in the system. If you do it that way, you build in almost a guaranteed disconnect between the research data through specimen as to what is actually being seen in the clinical context. So we invert that and say no specimen gets into data generation unless it's been reviewed and QC'd at the same level you would to make a diagnosis. So you at least bring those worlds close enough together that an observation here does begin to inform on the clinical reality. And so it goes all the way from, consent all the way through pathology, through standardized data generation. So the output is comparable, not only within tumor type, but across tumor types. Because we know, particularly going back to immunotherapy, there are common rules of the road. We're just beginning to understand them. But if you didn't collect the data from lung to breast to prostate to, adrenal cortical carcinoma the same way, then all you're measuring is the difference in the way you did the collection and the data generation, and not the difference in the biology of the disease. So it's hugely important to standardize these early steps in experimental design and sample collection and data generation to even begin to have a hope of having insight on the backside. So it was a real commitment on the institution's part to say the plumbing matters.
I mean, Andy, this point is great. I mean, I like where you bring your hands together. The observation, the measurement to the insight. And so in a digital world now, we're taking those measurements and understanding that we're going to connect that data to the insight. And then we keep the connection, which is this sort of way to move faster. When we get the insights dependent on this observation, let's get more of those observations. And we, rather than kind of just throwing it over the fence, tied together. And it's possible now with like digital fabric to really connect those two. And we've seen that activates a really exciting sort of clinical science discovery conversation in the organization. And that's been a really exciting several months for the past little while. We've been working together on it. It's very exciting.
And do you see, David, any kind of like short-term gains or biggest bang for your buck investments that you're making to really drive forward that goal?
I mean, a couple of great examples. It's really human, actually. It's not that digital. It's putting teams together that work collaboratively on data sets that are coming through the system, and working collaboratively to understand them, to name them consistently, putting the semantic frameworks around them so that we can really understand what does this observation mean relative to the next observation. That kind of data management groundwork is key. But what and everybody who does science or works in the technology would know that's important. But what I'm seeing is there are technological innovations now coming forward that can facilitate that. There are tools that are allowing us to start to keep track of that kind of information. And we've seen in a recent collaborations that, people put time into making sure the data is high quality and curated. A lot more people can work on that data if you do it in a structured way. And part of it's technology, part of it's culture. But it's a lot of teaming involved. That's been fantastic for the past several months.
Can imagine. And you have the plumbing in place, you get the culture right, you put the teams and processes in place, collect a huge amount of data information. Are we starting to get to a point where we can start to leverage machine learning and AI on that data, and especially historical data that you've been collecting over decades now, to start generate new insights and new discoveries in your field?
Well, I think this is one of the things on the technology side that it's a kind of a perfect storm. I mean, the ability to connect is great. The ability to measure so much more at a lower cost. Andy's demonstration of how we need to make these measurements in a controlled fashion, but feeding that into a human mind alone has its limits. Humans are pretty good at, weighing out maybe 4 or 5 different things and making a decision and understanding. We need computers to start to look for correlations across these observations, because if we wire all that together, it's very challenging for the human mind to start to bring the full scope. I mean, why would the patient who's on a diabetic therapy have a different outcome in response to a prostate cancer treatment? Like, why would that information way over there affect what's happening here? But yet it does. And so that scope, that variety of data to deal with it moving forward, we need to learn how to use computers to help us watch for those correlations. We won't get to causation. We'll talk to Andy about causation. Maybe the computers will say causation one day, but I think I'm going to lean on Andy more than the computers for a while. But I think this is key. I think the scaling needs the computational scaling as well. That's machine learning AI.
And have you seen any examples of this play out in terms of looking back on maybe some information using these technologies and saying,
Yeah,
okay,
there's a couple of really great domains like this. For example, imaging data, super rich, very complicated. There's been a lot of work recently and having computers look through it for textures and patterns, so-called radiomics, which is sort of like the features of an omics embedded in an image. And the reality is there's no way a human would look through all those images and look at that texture. The only way that works is through automation and the use of computers. And so just the pouring over of that data and finding potential that some of the texture in these images predict for differential outcome, that's been repeated, and there's still not in the clinical context yet, but there's some promise there. And more importantly, how do we connect those types of omics back over to the omics that Andy was talking about moments ago? So we're starting to see how the computers can really change the way we look across the data. And it's kind of interesting because we're normalizing the way we talk about data now, whether it's imaging data or a genomics data or a microbiome data. And this data starts to flow into what I see in the future as very common forms of pipelines for correlation analysis and so on and so forth.
I mean, I think there's an inherent sort of challenge in the retrospective data. And it wasn't collected for the purposes of going back and parsing it out to do machine learning. So there's this idea that if you get enough of it, you'll find the signal if it is actually real. And I think some of that is absolutely true. But it's the balance against that and saying, well, actually, the other thing we need to be thinking about are really high dimensional measurement prospective cohorts as well. That has the disadvantage of you don't have the long follow-up in the context of cancer patients. So it's trying to find the balance between those worlds and being able to bounce back and forth where you have really good certainty about the quality of that retrospective measurement. And there's certain areas where that's absolutely, tractable and doable. There's others where it's a little more difficult, but imaging is clearly one of the places. You have the images. You could go back and remeasure them if you want to. So I think there are places, and we've done some stuff in the lab looking at predictors of outcome in the context of certain subtypes of leukemia where, you can begin to sort of play in this space of machine learning and develop some, models that do reasonably good predictive value. They're not necessarily better than what's being used, but I think we're in the place now where we can begin to really dig in and understand where we're deficient in the retrospective data space and how we need to design better prospective studies or even just get more standardization into the pipes up front so they can do a much better job. And there's certain cancer types where I think we'll make headway sooner rather than later because of the outcome times. But I think it is that sort of trying to get the best of both worlds and getting the data in a common platform is one way to do that.
Another interesting piece there is that, people want to know where the data came from that led to the conclusion that's robust. And so this kind of connecting the dots piece that Andy talked about when he brought his hands together is becoming kind of the only way we're going to do work that anybody will believe in the future. And sort of industrializing that connection is key to understand where it came from and how did you get the conclusion and is it reproducible? And this is I think it's going to be table stakes for real clinical science and real science moving forward is to figure out how to connect those dots and keep those connections live. And I think it has a great promise because we'll use our data a lot more effectively moving forward because of that.
Definitely. And we're getting some great questions around this as well. And one of them is around the trajectory of where we are on this kind of curve. What do we look like on a year-to-year basis? Are we sort of making an accelerating progress in our leveraging of this data? What can we kind of expect going forward?
Do you want to take that one, Andy?
Yeah, I'll take a swing at it to start with. I think don't think there's anybody who would argue that, we are making larger and larger gains and progress for better therapies for patients. Nowhere near where we need to be. We don't have a therapy for every patient who walks in the door where we have good probability that it's going to work. But I think there are tremendous advances, which are distributed broadly across lots of different cancer types now, that I think give us hope that even blindly stumbling along as much as we have been, we've been able to get this far. And if we just open our eyes and get a little bit better focus, how much faster we'll be able to move. Mm-hmm. If you look at the complexity of the system you're dealing with, I mean, there is every argument that says we shouldn't be able to treat anything, as plastic as the genome of a cancer is, as evolved as the system can be. But we do a pretty decent job in a lot of different cancer types, even if we don't quite understand why. And it's the balance of understanding the why we are as good as we are and how we can be better and what we need to be doing in terms of therapeutic development, measurement, et cetera, the whole nexus of activities and variation in the system that I think will drive progress even faster. There's no huge technological barrier to any of it at the moment. You can sequence relatively cheaply. Computation has grown by leaps and bounds. So there's very little keeping us from doing these things other than sort of the organizational and cultural and resource will to a certain extent, in my view. It's preachy, but there we are.
Well, I totally agree. I think of us as we've been running along in first gear with the engine at 5,000 RPMs. And we're going, why aren't we going faster? Why aren't we going faster? When we can make this transformation, this shift to think about the data and thoughtful measurement and assembling, and we have interventions that work, as sequencing them and optimizing how we apply them, making full use of the information. That to me is like a gear shift. That's that process change, putting it all together, and then being much more directed in where we go. And for us, it's a cultural shift to go there, but at the same time, it's a process and technology shift to allow that to happen in an efficient way. And then things like automation kick in, and we can move faster. And that's the kind of switch we're excited about.
And I think, we have a lot of entrepreneurs in listening to us today. And I think we all look at the technology challenges and opportunities there, and you're saying, well, look, that's not really one of the main components. It's a lot of this cultural perspective and the cultural shift and all that. So that's something that is harder to perceive the importance and weight of it from outside of an institution versus inside. So maybe just give us a little bit more around that. What is the mindset within, it doesn't have to be MD Anderson specific, but just the industry of how they're looking at, the value of data unlocking and all these kind of aspects, and how does it have to evolve?
I mean, I'm happy to jump on that one. There is an interesting shift in collaboration approach paradigm. I mean, what we're seeing from an open data perspective is very interesting. These sort of FAIR principles. I don't know if you're familiar with the FAIR principles, but the F stands for findability. How do you find data that you could use to help answer your question? We don't have tools to do that. Within an institution, we wrestle with that. We're big. And how do we allow people to find data and work together on projects? But between institutions, the same problem exists. And what we're seeing is other institutions are realizing, hey, we need to go through this transformation. Because if we don't, you're not going to be leading. You're not going to be setting the pace for progress. And it's not just academic centers and the health centers. Industry is also just as interested at taking away the barriers that keep this from this and Andy's illustration. They want to understand how can we work more closely and more effectively while respecting all the issues around privacy and security and so on and so forth. How do we just get more efficient at making progress against this disease? And there's a real appetite for it. I've been really struck by it.
Yeah, and I would echo that. I mean, sort of in dealing, and I've moved back and forth between the basic science, if you will, world and the translational world a lot, like 1,000 times a day kind of thing. But what I can tell you is that if you begin to show value in terms of an insight into why the patient that a physician just treated may have responded or not responded, then that is the single biggest push on a culture change that I can potentially think of in the environment we work in. Mm-hmm. And that's what we have to be able to do. You have to be able to show value, and not just academic value, which has its own merits and value because it generates resource, but it has to eventually, and everything has to have a line of sight on how it will impact the ability of our colleagues to do their job, which is to take care of patients, but more importantly, to impact the patients themselves in terms of better outcomes. If you can begin to show value in that space, then the culture comes along. It may come kicking and screaming in some individuals, but it will come along. And I've been in my going on 9 years now at MD Anderson, I've never once sat in a room with somebody who said, I don't get what you're talking about. They thought it was very difficult to do, and they may or may not have a burning interest to get in deep and start digging ditches to make it happen. But everyone got the reasons we were trying to do what we were doing, and not just humoring me to get me out of their office most of the time.
Fantastic. So looking a bit, towards the frontier of where this is going or could go, and the value there, looking at predictive medicine, precision medicine, are we on a path where we can use computers to start to simulate, to project forward, and start to map out a patient's response probability, etc., all using this data? Is that where we're going? And if so, where are we on that path?
I mean, this is critical. I mean, when a patient sits down with the physician wants to be able to predict for them, given an intervention, what the outcome is going to be. And they want to help them choose one that's going to be as likely as successful as possible and with low toxicity. So that prediction problem is extremely important for so many dimensions. We'd love to be able to predict 100% cure every time as well. But that prediction problem is a real problem for many, many people every day in a cancer center. And so how do we use computers to do that? We can do sort of fitting-based approaches like AI machine learning paradigms, where we can look at the features as long as we know the outcomes and predict that this combination of features predicts this outcome. And that's a pretty classic, prediction paradigm. But there's also another bunch of work that's started, which I'm super enthusiastic as a physicist about, is that really true mechanistic models that start to use the full computing horsepower that we've been building with measurements like imaging and other measurements to start to generate models, like weather models, like not an almanac approach to figuring out what the weather is going to be, but actually take an image and based on that image, try to figure out what's going to happen to that tumor. Is it going to respond to the immunotherapy that Andy just talked about, or is it not going to respond? Because that's extremely helpful in making the right decision for that patient. So I'm convinced that detailed computational models is the way to go, like we use in the weather paradigm today, but for individual patients. To me, that is the exciting frontier.
Wow.
Yeah, no, I would agree. I mean, we're sort of we have guidelines now, national guidelines that if you present with, a stage 2 lung cancer with this minimal set of characteristics based on a large scale phase 3 trial, you should probably do this. And on a population scale, there's probabilities associated with, your likelihood of response. We need to get so much deeper than that in terms of the individual measurement on the individual patient and not, present group statistics. There's a 20% chance of response. Well, if you're the patient, it's not 20%. It's you will respond or you won't respond. And where do you sit in those various trajectories, I think is huge. And more appropriately, what sort of measurements do you need to have to say, if we measured at time 0 and time 1, do we learn something at time 1 that now says, well, actually, the trajectory we thought you were on isn't quite what we believed, and now we need to do something slightly different to bend the curves a little bit. So this ability to interrogate a dynamic system with the appropriate data to make the decisions and have, of course, the available therapies to sort of switch to, I think is that magic sort of combination of things we want to drive towards.
And that requires data and measurement.
Yeah.
And flowing
it's all
data,
about data and the measurements, yeah.
into computers, CPUs, GPUs,
Flashing
better
lights.
models.
And the flow of data is key. Sounds like the continuous flow and that dynamic nature of it is important. And what's interesting when we have our discussions together is around how much is happening outside of the institution instead of collecting this data on that patient. And when we were talking about this topic ahead, you guys are mentioning that potentially more measuring will happen outside of MD Anderson of its patients, more engagement, more data collection than within. So if that's the path that healthcare is on in terms of this sort of outside of the organization, what does that look like? How is it being collected? How's it being leveraged? And where's it going?
I mean, that's an exciting future, I think. I mean, people buy devices that make measurements of themselves all the time, Fitbits and iPhone watches, iWatches, I guess, all kinds of devices. And with time, I really believe that the amount of health information being collected outside the healthcare system will exceed easily the amount collected inside. And we need to figure out how to use that data for the benefit of the patient. And so for us, it talks about how do we collaborate with patients? How do we digitally engage with them? We've seen it already in the COVID context, people submitting their symptoms into apps and things because they know there's a collective benefit. But moving forward, how do we see people contributing their own information so that we can better guide their care? We see it in the diabetes paradigm already. In the cancer paradigm, I think we're going to want to know what was your outcomes, what were your symptoms, and help share that with us so we can include that in the calculation. And to do that sharing, we need new approaches, new technologies, and we have to really build a relationship from a patient engagement perspective, and the tools to allow that to happen. And that's just such an exciting frontier. I think it's an exciting future.
Yeah, I mean, and we've talked about it a lot, David, in the context of, moving away from this idea of patients as research subjects and patients as sort of research partners over a longer time scale. Mm-hmm. And, there's some nice examples where there's really interesting data emerging. We come back to the microbiome as an example where here you have an opportunity where you can actually self-manipulate that system and give patients agency in their own care, where they can change the dynamics of that system by changing their diet, exercise, and various other things. And I think we're moving into this era, which is really sort of interesting. I think will change how we think about long-term cancer care, where the patient is now a partner in their own care as opposed to somebody that comes in and has stuff done to them, so to speak. Mm-hmm. I think that would be huge as well.
Real partnership there, and I think one of the things that's pointed to as that gets a lot of interest, and part of these questions as well, is around liquid biopsies and how that can happen outside of the institution. So help us unpack a little bit of the hype and reality of that. Is it going to solve for a lot of the issues that maybe traditional biopsies couldn't pick up on heterogeneous natures of tumors, or etc.? And how do you view its role?
I'll take a stab at that one. I mean, certainly, you can learn a lot in the context of the characteristics of the tumor from a peripheral blood sample. That's been demonstrated, I think, without any question. The representativeness of that, of the entirety of the complexity of a disease in a patient, it's as good as place as any to go try to measure it. Certainly, going to interventional radiology and taking a single core from a single area of a single lesion in a patient who has 15 doesn't tell you about the heterogeneity of the system. I think there's still a need for some fairly controlled experiments to understand across tumor types and across stages of disease. I think in particular how well you're actually doing in the peripheral blood space, as an example. But I've no doubt that as we get better at measurements on smaller and smaller bits, if you will, a lot of what we do will be done from a peripheral blood draw. And I think that, again, changes the ability to measure over time in a way that, we just can't really approach with a significant number of different cancer types at the moment. Some lend themselves to longitudinal, if you will, sampling more than others, but the majority, you can't biopsy, but you can do blood draw as part of routine care when patients come in for screening, et cetera. So I think there's real hope, I think, and faith that it will play out, probably more so in some tumors than others, but I think will become a standard way we just measure tumor response and, overall organismal response, if you will, over time.
I think there's an interesting fusion there that's going to start to happen. I've seen many exciting proposals in this space recently where we take those circulating markers and we start to compile them with other measurements like imaging and start to put model-based views of that patient and their response together. And because it's not possible to make every measurement in an invasive way, that's just not feasible. So the circulating markers informing changes that can be observed through imaging. And this back and forth paradigm, I think it's extremely exciting, but it needs a model. It needs a model to be assembled. It needs a computational framework. And those will be substantial, computational activities. But this is where we work with what we can measure it in very high quality, and then use the model to try to interpolate and understand the impact for that patient. And I'm convinced this is this is a fantastic space moving forward.
Right, and I want to leave a lot of time for questions because we're getting plenty of them. One of the ones that people want to hear the most of is what are the technologies for furthering personalization that you each are personally excited about?
Andy, you first.
Well, I think that the ability to measure more and more from smaller and smaller samples, I think, is key. I mean, I'm grounded in traditional, so I did a PhD in pathology, so I think tissue, almost by definition. But the fact of the matter is, if you can do more and more with less and less, then you are able to access it more frequently and more larger groups of patients. And again, that becomes the key to having the insights to build the models, to be able to do the predictions. And so I think that space, the technological leaps we've made, and the ability to detect things with high veracity in smaller and smaller samples, do genomes on a drop of blood, those sorts of things, I think are all game changers. But the other game changer on the back end, I think, is getting back to the heterogeneity piece, is sort of the worldwide explosion, if you will, in understanding, variation writ broad across the planet. So now we have a framework to understand all the variation we're going to see in terms of 20,000, 30,000 genomes being sequenced in human cancers. There's a framework to understand the next patient who walks in the door in a way that, in the beginning of my career, we were flying utterly blind. We didn't have a genome. And now you can instantaneously, to my chagrin and my old man sort of pontificating to graduate students, you can know exactly the context of that within seconds. And I think that ability to have that reference writ large across the genome and across the planet in terms of cancer patients and variation populations is absolutely huge. And we're just beginning to scratch the surface of it in terms of this idea about how similar are you who walked in the door as a cancer patient with this suite of phenotypes to someone that we've seen and treated before. And I think that ability to rapidly iterate across that space is the thing that I think is really exciting in terms of that nexus of molecular measurement and computation and data.
I totally agree with Andy. And then the other side of the spectrum is the sort of how we're using technologies to really tune the intervention in a way that's way more controlled than in the past, and do it in a very consistent way. So, targeted and controlled placement of radiation to minimize toxicities. Those are, very, very powerful development of, proton therapies and the like. Just targeting to try to take out the variation in the intervention and to keep the toxicity down. And that kind of personalization, I think, is really powerful. Once again, heavy requirement for computational needs. But also, as the treatment changes, and this is what Andy, regardless of the kind of disease, how do we put the guidance frameworks in place? Not the spatial guidance, that's one part of it, but the temporal guidance frameworks in place. How do we respond to the fact that that patient's not responding? How do we map that through time? And that kind of navigation or guidance system, once these measures start to come up, we can tune and tweak intervention in a way that's highly optimized across modalities, which I think that coordination problem is very powerful from a personalization perspective.
And I want to come back just to the idea of a model, individual model for each patient, because I think that's really powerful notion. If you talk about what we've been talking about today, a continuous flow of data through continuous sampling, whether it's a blood draw or whether it's other things like a Fitbit, etc., whether it's inside the institution or outside of the institution, creating this model around that patient. This is a very complex undertaking to do. So two questions in this one, probably for you, David, is what does that look like from a technology perspective from your end? And then what are you going to do in-house versus engage the wider ecosystem around you in terms of partners, etc.? And how are you thinking about that?
We build off of the things that Andy talked about, putting the ability to acquire this data consistently in a robust fashion. And it has to be quantitative. It has to be certified. The data has to be the highest grade. The idea to think that it flows into a decision about a patient through an algorithm means that it has to be fantastic. So we have to put those elements in place. And then that flow of data through time, we know that every patient who presents, the time we decide to treat them, isn't exactly the same cancer that the next patient over has, let alone in its development in time or in its genesis, as Andy has highlighted. So calibrating that and getting that built, and then starting to put models in place, computational models, this is exciting new work that's happening. I think it's a great future, but thinking about the computational requirements to do that. And what we're doing is we're reaching out to strong computational groups, computational physics groups, large computing infrastructure. We have a collaboration with UT Austin and the Texas Advanced Computing Center and the Odin Institute, talking about how do we crack this nut? How do we bring substantial computational power and models to this problem? And so we have to partner. Industry partners, academic partners working differently. And one of the barriers there is just figuring out how to share data properly and manage data in that whole pipeline. So it's got a number of dimensions to it, but the partnerships we've seen in industry, they now skip complete prototype versions because the simulations for their products are so good out of the gate. And we have to learn from that and figure out how we can bring that into medicine.
Mm. And a lot of startups would love to partner with an institution like MD Anderson, start to show the value that they can do mining that data that they've developed. How do you interface with that ecosystem? How do you take advantage of that while mitigating, obviously, the risk and regulation on your end?
Well, one of the things we've been doing, and this has been quite exciting as part of our sort of rethinking how data flows, is put together a framework where it's pretty clear what the architecture is in the organization for data flow and the governance and framework for that and making decisions. We want to do that for our own agenda, but it also creates an opportunity to sort of click in different technologies. And those technologies can come from labs or they can come from companies where they bring a new algorithm. But if any company that's listening today has ever tried to work with a healthcare organization on accessing data, it's a nightmare. I know, I've been on the other side of that nightmare a couple of times, or both sides of that nightmare. And what we need to do is get rid of that. We need to make it so that it's possible to partner better. And so we've been working hard to start to build a structure that allows that to happen. It's not perfect yet, but we need to bring innovation to the coalface faster. And so we're committed to that, and getting our data organized and understood so that we can do it effectively and responsibly is key.
And is that structure something that's going to be in place in the near term? Is it a very heavy lift internally, or?
It's a heavy lift, but the culture is there, and so we've started. Not to say that everything that clicks in doesn't also click out after a while, but we believe this is the right way to go. And organizationally, institutionally, we're aligned on collaborating on data in a way that advances the mission.
That's great. That's great. Well, we're coming up on time, but one topic. So you're doing amazing work out of Houston, and then you've got a global network as well. How does it look like from an international perspective? What's the collaboration look like, within the US and then externally, internationally, to really make this a global effort?
Well, there's certainly long-standing examples around, in the academic sector of global-sized collaboration. When you look at the International Cancer Genome Consortium, that essentially that has changed the landscape of what we know about human cancer from a genomic context. So I think there's good examples that you can collaborate across, state, international, and latitudinal lines, and the sky not fall because bad things don't happen. I think there's a model out there that says, data sharing, which I think has some value. But I think what we've learned over the last number of years is purposeful collaboration actually will get us to the finish line faster. And we need to be really sort of, thoughtful and go after real questions. And there's an academic sector who will churn on data and understand things, develop new methods that can be used in the former scenario. So I think we have to learn how to empower both, but not kid ourselves that one gets you the other necessarily, particularly in something where every day matters in the context of things like cancer, where you can't wait for the flowers to grow. Sometimes you have to plant the damn garden and see what happens. And I think that's really important to sort of keep an eyeball on as well, in my view. Again, a bit preachy, but there we are.
It's perfect, Andy. I think this is key, and there is a future collaboration global with data that's coming, and organizations have to be prepared to participate in that. And technology, and using the technology to do it with precision and intent, is going to be key. the current sort of sharing data paradigm just doesn't have the precision necessary to have intentional, robust collaboration. So I see this as organizations have to kind of put the architecture in place procedurally, and then we need digital technologies that let us really collaborate efficiently and draw together and extract insights at a much higher pace. I think that's a huge opportunity from the tech sector perspective.
Indeed, indeed. And you know, with leaders such as yourself at major institutions thinking that way, I think we've got great hope in achieving that. So I think that's all we have time for, unfortunately, today. I know these are topics that everyone could talk to for hours. So thank you so much for your time, David and Andy, and your insights there. It's been an incredibly rich conversation. And thank you for joining us, and thanks for everyone who's joined as well.
Yeah, thanks for having us. And thanks everyone for the questions.
Amazing conversation. Thank you, Dr. Footshill, Dr. Jaffrey, and Daniel Darling for moderating such a great event. Thanks, Maddie, for making this happen.
Thanks everyone for joining, and thanks to our amazing panelists. Again, for links for the YouTube, which I'll be uploading the recording, check the chat, and please join us next Thursday at 11 a.m. for Taking Action with Sustainability in Our New World. See you guys then. Thanks for joining.
Right.
Bye.
Thank you.